My job is to tell you about the mechanobiology of molecules. The goal is to fill in some of the details related to adhesions and cell-cell attachments and how the molecules that are in those structures respond to mechanical forces. Okay, and my goals, this is really dim. Let's see if this one's better. Huh, nope. Okay, so the goals of this lecture are to provide some simple examples of how molecules mediate the sensing of mechanical stimuli. So if we're going to sense force and we're going to respond to force and we're going to change signaling in response to force, those molecules somehow have to feel the force. And so I'm going to tell you a few ways that these molecules do this. And I'm going to give you some very simple biochemistry, some very basic biochemistry that'll give you an idea of how one goes about thinking about mechanical load affecting something like an equilibrium constant. Thank you. All right. Okay, is that better? Yeah? Okay. And so then in the last bit, I'm going to briefly discuss the molecular motors that power force generation in the mechanobiological processes. So molecular motors, cytoskeletal motors, which you heard about from Erika Holzbauer, not only drive and power mechanobiology, they sense and adapt to mechanical load. And I'm going to provide you a very brief example of this, and I'm going to show you some structural changes that mediate this force sensing. Okay, so as you know, and as you've heard now through these weeks, is that there's a wide array of molecules that synergize to generate mechanical forces, produce power, and connect structural elements and also respond to signaling. So this is a movie that we acquired in my lab many years ago showing a cancer epithelial cell migrating up towards the upper right of the screen. And you can see this broad lamellipodium that's pushing forward, that is responding to mechanical load. Polymerization, actin, myosin are all working together to push this thing forward. We also have molecules at the back end of the cell that are pinching the cytoplasm, the back end of the nucleus, and pushing all of this stuff up. We also have molecules that are talking to each other between cells, these intercellular junctions, which you heard a bit from Rick Asoyan. Okay? And so not only do we have these molecules that are generating and pushing these forces, pushing the cells, but we also have molecules feeling the substrate, putting this cell on a genetic program in response to what it's feeling on the substrate. And so there's lots of molecules that are mediating these effects. And they include the matrix cell and cell-cell adhesion proteins, ion channels, and you're going to hear a lot about ion channels in the recorded lecture after mine. I'm only very going to briefly talk about them. Signaling proteins, so proteins that are responding to what other proteins are feeling and signaling to the nucleus. Scaffolding proteins, so these are proteins that connect all of these other important proteins and act as a signaling hub. Cytoskeletal filaments are polymerizing, driving, pushing this leading edge forward, and molecular motors are really the proteins that power mechanobiology. Okay, so how do all of these things work together? And what are the changes in these proteins that are able to mediate this force sensing? So you can sense force and respond to force in different ways. So the cells do this. They use different processes to do this. One is to change the lifetimes of intramolecular bonds. So mechanical load can feel, so the proteins can feel mechanical load and change how long they stay attached to each other. Okay, and you've been introduced, I'm sure, to the focal adhesion more than once. So this is this adhesion complex that binds to the extracellular matrix, strengthens upon mechanical load, and talks to the nucleus. So in this one structure, this very complicated structure, there are different types of adhesions, different types of intermolecular bonds that are formed between these various components. And generally, when mechanobiologists talk about adhesive bonds, they talk about them in two ways. They talk about them as a slip bond, and they talk about them as a catch bond. So have you talked about this yet? No? Okay. So a slip bond is a very intuitive kind of bond. So if you have two proteins that are attached to each other, you can measure their lifetime. So you can say, what's the average amount of time these two proteins stay together before detaching from each other? Okay? And if you look at the average bond lifetime as a function of force, a slip bond has shorter lifetimes as you pull on it. Okay? So it's kind of intuitive. The harder I pull on it, the faster it'll come apart. Okay? So that's a slip bond. Now there's another type of bond that's very important that I'll show you a couple examples of, and that's called a catch bond. So again, here's the catch bond, and if we look at the average bond lifetime, so how long these two things stay attached, as a function of mechanical load, under a certain regime, this bond lifetime will increase. So mechanical load will actually increase how long that bond interacts. Yeah, question?
So I'm part of the RET program. So in a high school setting, when you say non-covalent bond, and we're presenting bonds, chemical bond, as either covalent, ionic, or hydrogen, what category would the slip bond?
So a non-covalent bond is basically anything that's not covalent, right? So in a protein-protein interaction, you have electrostatic, these ionic interactions, as well as hydrophobic interactions, okay? So a protein-protein interaction could be quite complicated. That includes multiple, you got these ionic, van der Waals, hydrophobic, there's these very complicated interactions. So this would be anything that's not, just not a tight covalent interaction. Okay, does that answer your question? So it could be a combination of every other thing. Okay, so if you look at this catch bond, as mechanical force increases, you increase the lifetime. So this could be very useful when you're thinking about strengthening the presence of a complex. So you've heard about focal adhesions, and you've heard about focal adhesions maturing, maybe you've heard about that, yeah? So as a cell attaches to its substrate, or as cell-cell interactions increase, these bonds will strengthen with mechanical load, and part of that is due to this catch bond behavior. Catch bonds, after a certain load, will become a slip bond, and the strong bond will be overpowered by force. Okay, any questions about that? There's also a bond called an ideal bond. And an ideal bond is defined as a bond that's completely not sensitive to force. Okay. But I think ideal bonds are probably just looking at a certain bond over very small, or it's very load insensitive. I think most bonds will have some kind of load sensitivity. Okay, so these are mechanical load affecting the lifetimes of bonds. Mechanical load also induces conformational changes in molecules that affect signaling. So if you have an extracellular matrix and you have integrins and some kind of adhesion that's attaching to the actin cytoskeleton, mechanical load can change the shape of that molecule and reveal other binding sites. Okay? And so these other binding sites could reveal some protein that you could bind that's important for localization of other proteins. It can open up a regulatory site, like a phosphorylation site, that will subsequently allow other proteins to bind. And I'll show you examples of both of these in just a few seconds. Okay? Any questions about this? Cells also sense and respond to mechanical load by affecting the open probability of a mechanosensitive ion channel. So changes in membrane tension or changes in membrane cytoskeletal interactions can affect the open probability of a channel. So I think most of you know that ion channels are extremely important signaling molecules that change potentials at the membrane or allow ions to flow that are important for signaling. Now, you're going to hear a lot about this in the recorded lecture after mine, so I'm not going to talk about it in detail. I will introduce it again in just a few minutes. Okay. Cytoskeletal proteins also respond to force. So in the case of actin polymerization, that's important for moving something like the periphery of a cell membrane. Mechanical load will affect the ability of this polymer to add subunits and will affect the rate at which force can be generated. Mechanical load also affects molecular motors. So this is an actin filament bound to a myosin molecule, and the myosin's undergoing its ATPase cycle. And this ATPase cycle is responsible for generating mechanical work. Load that resists this power stroke affects the ATPase kinetics, and I'll show you about that. I'll show you some details related to that at the end of the lecture. Okay. All right, so first let's just talk about a couple of molecules in focal adhesions. So again, we have these really important structures that cells have that attach them to the extracellular matrix. They're attached to these things called integrins, these important transmembrane proteins called integrins that bind several other proteins that ultimately connect to the actin cytoskeleton, which binds to myosin, and they're even linked through various mechanical linkages to the nucleus, and they affect gene expression. Cells can sense applied forces and the properties of the extracellular membrane through these adhesions. Okay, they can sense rigidity, they can sense topography, and they can sense the anisotropy or the orientation of the filaments in the extracellular matrix. Cells change their shape, size, and orientation and migration in response to these mechanical factors. So what goes on inside of the cell is directly dependent on what the cell is feeling through these focal adhesions, through these integrins. Okay, that should be review. Is that review? Yes? Okay. So focal adhesions develop along the direction of applied force, and relaxing the force causes disassembly of the adhesions. So these focal adhesions are very important mechanosensors. They're transmitting this load to the extracellular matrix, and they're feeling the mechanical properties of that extracellular matrix. So what are the properties of these proteins in the focal adhesions that allow this to happen? So now this is a very, very, very complicated machine. And there's lots of proteins that respond to force, and I think we only are just beginning to understand these proteins. But a couple of them are pretty well characterized, and I'm just going to give you a very brief introduction to some of the really important ones. So this is now a really famous picture of what the arrangement of proteins in a focal adhesion. And so in blue here you have the extracellular matrix, and here in red we have these integrins. So these are these transmembrane proteins that bind to the extracellular matrix. Attached to the integrins, you have an array of really interesting proteins that ultimately connect to the actin cytoskeleton. Okay, and so we have these proteins called paxin, Talen, vinculin, zixin, VASP, alpha-actinin. All of these proteins in some way respond to mechanical load. Okay, so first what I'm going to tell you something about is this protein called Talen, and how this protein Talen responds to mechanical load. Okay? In particular, how Talen affects the recruitment of a protein called vinculin, which is also mechanoresponsive. Okay? So there was this paper several years ago now from Michael Schietz's lab where they were looking at the mechanical load on a portion of the Talen molecule. This portion of the Talen molecule is called the rod portion. And so this is the high-resolution structure of Talen. And one of the things that the field had thought is that if this molecule is under mechanical load, it will stretch and reveal a binding site for this protein called vinculin. And this is vinculin in yellow, and in this alpha helix right here, that's the part of Talen that binds. And so what the Sheets lab showed was that pulling on this molecule reveals this site that allows vinculin to bind. Okay, so this is how this Talen protein is able to sense force. It senses force by undergoing this large conformational change. So it's even more complicated than that. So here's another cartoon showing the extracellular matrix. Here are these integrins that are going through the membrane, and here's Talen in green. And so what you see is that when there's mechanical load between the extracellular matrix and the actin, okay, the actin pulls on that Talen, and that Talen opens up, allowing this protein, which was cytoplasmic, to now bind to the vinculin, which now binds the Talen. And when vinculin binds, it can now cross-link actin filaments, and this binding now affects a protein even further downstream called zixin, which was binding a protein called alpha-actinin, which now comes off. Okay, so that's a lot of stuff. And no one here is asking you right now to memorize each one of these components in this pathway. But what I'm pointing out is that just this mechanical load on this protein now starts to recruit other proteins and sends other proteins off to go do some signaling. Okay, it's really elegant. Yeah, question?
So where's the pulling force in a cell, not specifically this, where would a pulling force come from, inner cellular pulling force come from? Wouldn't you expect the pulling force to be from the matrix?
So where does the pulling force come from, right? So ultimately, almost all pulling forces are intracellularly that attach to the adhesions are through myosin and actin, okay? And so this actin right here is attached to myosin molecular motors. These myosin molecular motors cause contraction and pull on that actin, which then pull on that adhesion. But the pulling can come from this side too, right? So if gravity is pulling on this part of the molecule, then this will have to respond. The inside will have to respond. Or if another cell is attached to the matrix and another cell is pulling on that matrix, this cell will feel those pulling forces.
And so if you have a pulling force from an extracellular pulling force, then would you expect the Talen to behave similarly to how it's behaving?
Yeah, so the Talen doesn't know which direction it's being pulled. So it's, if you have a spring in series and you pull on one side versus pulling on the other side, it doesn't care. That spring is still going to stretch. However, okay, so if there are other proteins that react prior to that Talen interaction, you could set off a cascade of conformational changes here before you set them off here. So there could be anisotropy in the system. Yeah. Okay, any other questions? Okay. Okay, so this is a mechanical change in a protein that causes the recruitment of one protein and reinforces this adhesion complex. Now, there's another example of these types of proteins that undergo a conformational change under mechanical load. But in this case, this protein, when it's stretched, can reveal a phosphorylation site. So this protein is P130Cas. This again is from Mike Sheets' lab. And this is an important scaffolding protein. In cells that aren't under mechanical load, this protein, this P130Cas, is largely cytoplasmic. Okay? And so there's two domains on this protein. that bind on either side to other proteins. And when this protein is bound and its attached proteins are under mechanical load, the protein opens up and it can now bind a kinase, which then phosphorylates the protein, which then can recruit other signaling proteins. So after mechanical stretching, the cast substrate domain unfolds, exposes a phosphorylation site that's phosphorylated by Circ family kinases, and then this subsequently pulls in lots of other proteins that are important for signaling. Okay? Again here, my goal isn't for you to remember all of these different types of proteins, but it's to show you another example of how mechanical load can generate an intracellular effect. Okay? And in this case, it's opening up a phosphorylation site, which results in very important signaling pathways. Okay, so this isn't all magic, right? So there's some real biochemistry and there's some real physics here, and you can measure these things. So we as mechanobiology scientists that are interested in the engineering of these proteins and how they respond specifically to mechanical force, which will ultimately allow us to model these processes, we can actually predict very specific properties of these molecules and how they will be affected by mechanical load. And so I'm going to show you a very oversimplified way of thinking about this. Okay, and it's good for high school too. Okay. So let's say we have so this protein, let's take this protein for example. We've got a protein that's got these adapters on either side. Then when you pull on it, it stretches this part and allows it, by stretching it open, reveals a binding site or a phosphorylation site. Can we predict how mechanical load will actually affect the probability of this being opened or closed? So let's take this really simple case here. So let's say this is our protein. Here it's in one state, here it's another state. So let's say this is the closed state and this is the open state. Okay? And without mechanical load, it's an equilibrium, right? Going back and forth. Right? And everyone remembers from freshman chemistry that you can calculate an equilibrium constant. So we can know what the probability of it is being in the open versus the closed. Right? And so let's say most of the time it's this closed state, and only very rarely is it open. So we can calculate something called the Gibbs free energy, this delta G, okay, which you've all seen. I know you've all seen because some of you probably teach it, and I think everyone's had basic chemistry. So you can relate some kind of energy to this equilibrium constant. Right? And you can even rearrange this equation to calculate the equilibrium constant from if you know this delta G. Okay, so that should all be review, right? Everyone got that? Okay. So our question is, how does mechanical load affect this equilibrium constant? So for example, this protein, it's in this closed state and it's in this open state, right? But there is some probability, even in the absence of mechanical load, that it's going to be open. But it's going to be a very small probability, which means this equilibrium constant will be very, very, very small. Okay? Got it? All right. Okay. So now how can we think about mechanical load here? Okay, so let's just say we have this one protein that's in this one state. And all we do is we translate its position. Right? So it's still the same protein, but we moved it to a new place. That took some energy. It took some work, force times distance. Right? Okay? And so the difference between this one and this one is simply the mechanical work that we put into moving it from one position to the other. Okay, everyone get that? Yeah? Force times distance, just the amount of work that it took to move it from one to the other. So now we can take this simple situation and apply it to this open to closed situation that we just talked about. So here we have E1 and E2. This is the closed and this is the open. And let's say we can now put mechanical load on this in order to pull it open. Okay? Force times distance, it took some work to pull it open. And so now the delta G isn't only this force times distance. It's also the delta G that we originally had from this equilibrium, right? So this equilibrium existed at steady state. By putting force on the molecule, we can now change the total amount of energy between these two states. Okay? You get that? So the amount of energy in our new steady state is the amount of work it took to pull it and the amount of energy in that original equilibrium. Okay? So now you can do some rearranging. So this is just these equations. There's nothing new here. And we can say this equilibrium constant is just this Gibbs free energy. But remember, now the energy isn't only that delta G. It's the delta G plus that force times the distance it took to pull that thing open. Right? And so now we can actually calculate the equilibrium constant, E2 divided by E1. We can rearrange these things. And it equals the equilibrium constant in the absence of force times this exponent of the force times distance times this thermal energy factor. Okay. So now we can do some really simple kind of back of the envelope calculations. So let's say this equilibrium constant was 0.1, right? So just very rarely we were here, okay? Most of the time we were here, right? Say it's 1 to 10, that would be 0.1, okay? So now let's do something. Let's say that length change for opening is 4 nanometers. The equilibrium constant in the absence of load was 0.1. How does force change the equilibrium constant? So now if we put on force, this is piconewtons, and we start to increase the amount of force, we can calculate how much we can change that equilibrium constant. So with just 4 piconewtons of load, we can make the equilibrium constant 6, where before it was 0.1. So we can greatly increase the probability of being in that open state. Okay, so this is very much an oversimplification for many of these processes. But basically, this is the root of how mechanics affects equilibrium constants. How mechanics affects that open to closed probability of that Cas protein. Okay? Any questions about that? Okay. So these are good numbers to remember. So if you're ever looking for relating piconewtons to KT, things to joules, just come back and look at this. Just find this. Okay? All right. So we also have important proteins called adherence junctions. Important protein complexes called adherence junctions, where before I was talking about focal adhesions, which allow cells to attach to the extracellular matrix. Adherence junctions are these complexes where cells attach to each other. Within tissues and organs where distinct cell types coexist, these contacts serve as external cues. This is how cells find each other. These are how cells put mechanical loads on each other, relevant to each other, without using the extracellular matrix. Mechanical loading at cell-cell and cell matrix junctions is critical for proper cell function and fate. Force transmission at adherence junctions is the driving force of multi-cell migration and the formation of tubes during development. Okay, so a lot of times we, as scientists, just consider single cells. We think about these cells binding to the matrix, generating force, right? But in a complex organism, these cells talk to each other, they interact with each other, and they have to move together. And they have to not only move together, they have to interact with a substantial amount of signaling machinery. So this is a cell-cell junction, and mechanical load on this cell-cell junction impacts an enormous signaling pathway, and this signaling pathway impacts this cell-cell junction. So what's an example of these adhesion complexes? Well, during tube formation, neural tube formation, epithelial folding. So these are epithelial cells, and here in yellow you have this ring of actin. Okay? When this ring of actin contracts, you get a folding of the epithelium to form these tubes. And one of the most beautiful examples is in Drosophila ectoderm contraction. So this movie is a developing Drosophila larvae. And so you can see those are all the cells inside the larva, and these are the epithelial cells. And so these cells are coordinating their movements. So you're looking at the outside, and these epithelial cells are zippering up, enclosing those internal cells. And these cells that are zippering up are feeling this mechanical load, moving together and responding to the adhesion to each other. Okay? So again, we have proteins that are responding to this mechanical load, signaling to the cell. And there's a couple of proteins, a couple of papers that have been really nice in the description of some of these, the mechanics of some of these proteins involved in these cell-cell adhesions. So this is from Alex Dunn's lab from a few years ago now. And here's one cell. Here's another cell on the top. And between the cells, we have these proteins called cadherins. So remember, the cells that bound to the extracellular matrix were called integrins. These are called cadherins. And these cadherins interact with each other and talk to the inside of the cell. So attached to the cadherins, you have these proteins called catenins, okay? And these catenin proteins interact, again, with that important actin cytoskeleton. So remember, I told you about these slip bonds and these catch bonds. And so one of the questions in the field was, do these proteins in these adherence junctions have slip or catch bond-like behavior? This is an electron micrograph showing a cell-cell junction. You can see these linear things in here? Those are actin filaments, and these actin filaments are ultimately connected to regions where these cells interact with each other. So the Dunn lab asked about the mechanics of these molecules with actin under mechanical load. And so they did a very clever experiment where they purified the key proteins in series with these cadherins and the actin filaments. And these included a portion of the cadherin, which they attached to a bead, you can see that in green, beta-catenin and alpha-catenin. So they generated this protein complex and bound it to a bead. And then what they did is they took a single actin filament and they attached a single actin filament between two beads, which they held in an optical trap. Have you guys seen an optical trap yet? Laser tweezers? You have? Okay. So for those of you that are shaking your head no, an optical trap is a very, very wonderful instrument that allows you to manipulate refractal particles with light. Okay, for some reason this isn't working. So you can put an infrared beam through a microscope, hold a refractal particle, in this case a bead, in the light, and using light, like a tractor beam from Star Trek, you can move these particles around. And one of the real advantages of this instrument is you not only can move these particles around, but when this bead that you're holding in the light is moved out of the center of the trap, there's a restoring force that acts as a spring. So this spring acts like a linear spring, just like Hooke's law. And if you know the spring constant, you can calculate how much force is on this bead by how far it's moved out of the center of the trap. Okay? And so what the Dunn lab did was took an actin filament, held it in two traps, brought the actin filament into contact with this protein complex, and then moved the stage and put mechanical load on the complex and asked, how long does it stay attached to the actin when it's under mechanical load? It's a very elegant experiment looking at these mechanical forces at the single molecule level. And I'm not going to go through the details, but they looked at the mean lifetime of this interaction as a function of mechanical load on that complex. And what they found was this complex acts like a catch bond. So at these forces between 0 and 10 piconewtons, the lifetime of the attachment increases with the amount of force that you put on the complex. Okay? As soon as you go beyond 10 piconewtons, the lifetime starts to decrease and shows a slip bond behavior. Right? And so they were able to model this. I'm not going to go into the details, but they say that this protein complex has a weak interaction and a strong interaction. And mechanical load moves this equilibrium constant from the weak to the strong, so more of these proteins stay attached longer to the actin filament when they're under load before they detach and become unbound. Okay? So just the inherent physical properties of these individual molecules give the complex mechanosensing behavior. The cadherin complex gets stronger under load. You can reinforce these interactions by putting mechanical load on the complex. Okay? There are also interactions through selectin-ligand bonds. And these are really important for tethering and rolling of, for example, leukocytes on vascular surfaces. So this blood cell leukocyte, as it binds to endothelial cells in the blood vessel, they will bind and they'll roll, and when they get activated, they'll invade. Okay? And they can do their infection-fighting job as they get through these endothelial cells. So when these cells bind to this endothelial layer, they have to adhere. And there's quite a bit of mechanical load on these cells after they adhere due to the hydrodynamic flow of the blood in the vessel. Okay? And so these interactions have also been investigated using single molecule techniques. And again, they wanted to know what the basic biophysical properties of these interactions are. And instead of using the laser trap, they used an atomic force microscope, which uses a cantilever, which basically taps the surface. And this cantilever gets pulled away from the surface. And since you know the spring constant of the cantilever, as it deflects, you can measure the amount of force on it. So this is this cantilever. Here is this surface that they're measuring adhesion to. And if you just look at the tip, this is the tip of that cantilever. They bind to these selectin molecules, these molecules that bind the selectin, which are covered on the surface. And they can compress this tip against, and they can pull it away, and they can measure force. And again, they see these molecules have catch bond-like behaviors. Okay, so I'm not going to dwell on this anymore. Okay, and so in the next lecture, you're going to hear a lot about mechanosensitive ion channels. So not only do we have these molecules that are very important for these adhesion complexes, just within the membrane itself, as a membrane is getting pulled, as the cell is feeling osmotic stresses, these mechanosensitive ion channels react to these forces. And I'm not going to dwell on them because you're going to hear about them later. But just to be complete, in prokaryotes, they act as emergency valves for osmotic stress. In plants, they participate in touch, gravity, osmotic pressure, and other developmental events. In animal cells, they're just so incredibly important in the nervous system. In ordinary cells, they play roles in regulating shape. We can hear because of mechanosensitive ion channels. We can sense gravity. We can hold our balance because of mechanosensitive ion channels. But again, you're going to hear about that later, okay, from Angela Schlegel. Okay. So in the last few minutes, I just want to focus on molecular motors and the force sensing by molecular motors. So from Erica Holzbauer, you heard all about these molecular motors that bind to the microtubules. Did she talk at all about actin-based motors? Or did she only talk about microtubule? Actin? Just microtubules? Okay. So these microtubule motors, of course, are important for the transport of organelles, for example, in neurons, but in all cells. But we have actin-based motors like myosin and myosin 2, which is important for contraction of your smooth cardiac and skeletal muscles, as well as contraction inside almost all cells. And you have the myosin 5 molecules that act very much like microtubule-based motors in their movement of particles and polarization of these particles inside of the cell. So mechanobiology is powered by these motors, and these motors respond to force. So even in those adhesion complexes we're talking about, and these conformational changes in these proteins that set off signaling events, the myosin motors themselves adapt their power outputs in response to mechanical load. Okay, and so myosins, we've got a lot of them. We have 38 different myosin genes in humans, and they do everything from power motions, power the myosin type of motions that you've been talking about at adhesions. They're important for moving organelles around, giving organelles their shape. They're important for hearing. They're important for cell migration. But in order to do that, in order for all of these 38 myosins to do this wide range of cellular processes, they have very different kinetic outputs. Some are really fast, some are really slow. We wouldn't want to be holding our posture with the same myosins that we use for sprinting. Right? So these myosins are biochemically and mechanically tuned for specific physiological processes. And that includes how they respond to mechanical load. So here's an actin filament. Here's a myosin molecule. This is, in this case, a myosin 2 molecule. And here you can see this myosin 2 molecule undergoing conformational changes in response to ATP binding in its active site. ATP binds, it gets hydrolyzed to ADP-PI. You get a reverse power stroke of the myosin. Myosin rebinds, releases phosphate, and then undergoes its power stroke and causes actin to slide. Okay? So that's how myosins work. All myosins do that. Some do it at 1,000 times per second. Some do it at 0.1 times per second. Okay? So there's this huge array of what they do. But they also sense force. So cytoskeletal motors, kinesins, some myosins, act as catch bonds. So this is basically the ATPase cycle I just showed you. I'm not going to take you through it in detail. But what we know is that the myosin undergoes these cycles between being bound to the actin, okay, and generating force, and detaching from actin, okay? And the myosin has to detach from actin as part of its cycle in order to reset its power stroke. Okay? So now when there's mechanical load on this myosin, the myosin acts like a catch bond and stays attached to the actin for longer than if it doesn't have load. So how does it do that? Well, myosins have a force-sensitive transition. So myosin, so this is the myosin's lever arm. When it releases phosphate and undergoes its power stroke, it then has another rotation of its lever. And this second rotation of its lever accompanies ADP release. Okay? If there's mechanical load on this myosin's lever, it can't go forward and can't complete the ATPase cycle. It gets stuck in this strongly bound state. Okay? And so we now know what these states look like, and we can actually look at the rotation of that lever arm and correlate with what's going on in its active site. Okay? Not going to take you through the details, but I just want to tell you that this actin detachment rate, this is now a rate, not a lifetime. The actin detachment rate slows as a function of force that resists its power stroke. So I told you about those previous catch bonds, right? This catch bond is different. This catch bond is affecting the ATPase cycle of the myosin. It's holding the myosin in a state where it binds strongly to actin, rather than a catch bond that's changing, the bond, which just simply increases its lifetime. In the absence of ATPase cycle, this myosin would act as a slip bond. Okay, so the details aren't important. I just want to point out these differences to you. So if you look at the actin attachment lifetime as a function of force for different myosins, you can see these orders of magnitude differences in the ATPase cycling. And this translates to very different power outputs for different myosins. So as mechanobiologists, when you're talking about power outputs of myosin, or myosins being recruited to a focal adhesion, or myosins participating in the migration of a cell, you need to realize that these molecular motors are not all the same, and they have very different responses to force. So if you're modeling myosin in a focal adhesion, you need to know its force dependence before you really have a good idea of how its kinetics play into the whole mechanobiology of the system. Okay, so with that, I've used up all my time, and I'll stop. Yes?
You mentioned here, so when the actin is under tension,
Yeah.
it's increase the attachment time.
Yes.
So that's effectively decreasing the actual power of the motor.
So the question is, as you put mechanical load on a myosin and you slow it down, you're decreasing the amount of power, right? But remember, if there's no force in the system, no work is being done anyway. And so what you see, if you actually, so it's complicated because it's related to that, to the force times distance as well as how fast it goes through that cycle. So as you increase the force and you don't decrease the kinetics, the power is going to increase. And so if you look at these various myosins, if you calculate the power output as a function of force that's resisting the power stroke, you can see that some myosins peter out very quickly, where some, like the ones that are pumping your heart, don't. So even though they have these resisting mechanical loads, they continue to generate power. So you just have to do the calculation. So your intuition is right. Lots of mechanical load when this thing is completely stalled and it's not undergoing its power stroke, there's no power output. But there's a regime before that in which there is power being generated.
And that would,
Yeah. Yes?
So you're talking about the conformational genes with the Talen and Adhesion.
Yes.
So do you think it'll, like, depending on the change in the substrate, the pulling of the Talen will change so that the conformational change between the two, there be like a weaker interaction between the two?
So the question is, if you change the substrate properties and you're looking at the Talen vinculin interaction, how is that related to the properties of that substrate? That's a really great question, and I think that's something that people are really trying to figure out. So if the extracellular surface is very stiff, any force that's going to be generated is going to be felt very quickly by those proteins, so they'll open up sooner. If it's a viscoelastic substrate and you're pulling on these elements and they're sliding and the force is being relieved, then now you've got kinetics. So how fast do they open and how fast do they close after they're open? So I think it's a good question. The simple-minded answer is if it's a really stiff substrate, you'll open it up right away, right? And if it's not, you won't. But it's actually more complicated than that because of the kinetics involved. And the amount of proteins being recruited. Okay, so I think we better move on to the next, to the recorded part of the